said something was. Or was it this morning? Oh, Carly, well, it's actually like almost done. Spells here and there.
Just go to sleep.
I don't know. It's like a good in a group together. What's our plan? She told me your plan.
It's just, no, but it's funny, it's easy. I don't do it hours, so it's all.
I have to learn a song.
We all do. It's gonna be so easy. It's gonna be funny though. Yeah. You can do like 2 seconds.
Okay.
Don't share it anyway.
Share.
I was not talking about you. Yeah. No, I just need probably already walked in.
My dad ordered them. Didn't even. My style. Yeah. Maybe we don't. Yeah, I just requested them if you want.
Feel like any air. Well, happy Friday, everyone. What does it say? February 20th? Is that right? Yeah. Yeah, I need to put in a work order because there's like no airflow. And one fun study I had a student do one year was, this was actually during COVID. We actually look at air circulation in like classrooms and other places. And you can actually measure airflow by looking at CO2 exchange because everyone's breathing, everyone's exhaling. And like using a carbon dioxide sensor, you can actually measure the amount of CO2 that increases in a room like this. And then you can, then after everyone leaves, you can then actually measure the CO2 levels again and see how long does it take for the levels to decrease the normal levels. And then there's actually some fairly complex formulas that you can use to actually measure air exchange. And then you can relate that to like airborne diseases. And there's actually some formulas where you could actually calculate, like if you had one person in the room that was like infected with COVID, what were the probability values of actually everyone else getting infected, so you can actually do those kind of fun experiments. So that's what it's kind of reminded me of this kind of, stagnant air that we have in here. So the message is that it's good to be in a room where there's ample airflow. All right, talking about air. That brings us to this slide here. So remember that what's the topic of chapter 11? Who can remember the general title of the chapter? Anyone? What's the title for chapter 11? Diversity of bacteria, that's right. So last time we started learning about metabolic diversity. So different ways that microorganisms can obtain energy, for example. And we started off by looking at some of those that live in an oxygen-deprived environment. So what's another word for lack of oxygen?
Anaerobic.
Anaerobic. Very good. So we had anaerobic, and another synonym for that is anoxygenic. So we saw some fun examples of that, and then the idea was that scientists believe that the planet Earth originally started or existed as an anaerobic environment where there wasn't oxygen. But then things changed, and they changed because of the introduction of these oxygenic phototrophs. So how long ago was that? Well, it's estimated that they were introduced onto this planet about 3 billion years ago. So just for fun, let's see if we can figure out what 3 billion looks like. Okay, so I'll start off with the number 3 on this whiteboard. And you tell me how many zeros to add to make 3 billion. Am I there? Okay. How many? 7? Let's see if I'm counting the number of zeros correctly. 1, 2, 3, 4, 5, 6, 7. Does that look right? How about that? I'll put some comments in there. Does that look about right? Yeah, so that's a lot of zeros if you think about it, right? And that tells you just how long ago 3 billion years ago was. Okay, so these organisms, somehow developed on the planet and started to produce oxygen, by photosynthesis, and changed the atmosphere, to where it is today. These organisms are referred to as primary producers. So what does that mean? Primary producer. Producer of what? Of energy, that's right. And so the idea is that, think about the food chain or food web, how these organisms are able to take light energy, from the sun, and convert that into chemical energy in the form of these organic compounds, which are actually carbohydrates. And then they are called primary producers because they are consumed by other organisms, like ourselves, other animals, and even other kind of microorganisms. So by being a primary producer, they help introduce energy into our planet. Okay, let's move on. All right, let's look at some examples of these oxygenic phototropes. And at the top of the slide, we have the term or word cyanobacteria. So cyanobacteria refers to a group of these photosynthetic microorganisms. And initially, they were known by another name. They were known as blue-green algae. So, wait, bacteria? Are they the same as algae? No, they're really different, right? Because algae, are algae prokaryotes or eukaryotes? They're eukaryotes, whereas bacteria are prokaryotes. So those are very different, right? And yet, people were confused, right? So you may have seen something similar to this photo at the very top right, where we see, a lot of growth of the cyanobacteria at the surface of fresh water. So it could be in a pond or a lake or near the edge of a stream. And these kind of explosive growths are referred to as a bloom. So you probably heard of that in the context of algae, but the same can be true for cyanobacteria. So sometimes the color isn't as dark green as that. Sometimes it's kind of I'm trying to think of there's a term for the bluish green. Help me out with the term. Teal. Thank you. I'm thinking chartreuse. Have you heard of chartreuse before? Okay. And you can kind of see how at the top where this coloration is on the surface of the water, and sometimes people think that it's similar to if you were to spill paint on water and oil-based paint and how that kind of floats at the top. So it has a similar kind of a weird appearance. And then you can kind of see this person's putting their gloved hand in the water. Why glove? Well, I don't want to touch that, but partly it's because of these toxins, these cyanotoxins that are released by the cyanobacteria that are actually harmful to humans and other animals. So that's one of the reasons why people are concerned when there are these kind of blooms in our local lakes and ponds. Why do we see these kind of explosive growths in the summertime? Any thoughts about that?
Sunlight?
More sunlight. Good. Warmer temperatures. And what kind of fuels? What kind of food fuels this kind of growth? What kind of nutrients fuel the growth? Fertilizers, so nitrogen-rich fertilizers. So you may have heard that when it rains, water runs off the surface of soils and can carry with it fertilizers into our water streams. Okay? And that basically fuels microbial reproduction, growth. Well, here's one example of a cyanobacteria. It's called Anabaena. So you have Anabaena, and you can see how, unlike a lot of bacteria, this one actually forms chains. It forms chains of cells. I can't remember if any of you have actually observed that your bacteria forms arrangements like with chains or not. Does anyone have bacteria like that? Because I remember I've seen a number of you have clusters. You have tetrads. I've seen those where you have 4 of them joined together. But I can't remember if any of you have chains or not. Anyone remember? Okay.
I had a few.
You think you had some?
I think so, yeah.
Okay, we'll have to look at that one again. But Anabaena forms chains. So, in a way, you can kind of imagine people would get kind of confused and think, oh, this is an alga, right? Because it forms these chains. But this one is of interest, though, because it forms this specialized and large cell over here in the center of this frame called a heterocyst. A heterocyst. And that's of interest because it can actually, what we call, fix nitrogen. So taking nitrogen gas and converting that into nitrates and nitrites. And because of that, some people actually use Anabaena as a source of fertilizer. They actually grow it up, harvest it, pull it out of the water, and then spread it out on the farmland or gardens as a natural form of fertilizer. All right, let's move on to a different kind of aerobic microbe. This one's referred to as a chemolithotroph. So chemolitho means that it's using an inorganic chemical as an energy source. So in this example, the inorganic element is sulfur. Hey, we've seen that before. We've seen sulfur bacteria before, but this one is aerobic versus anaerobic. So it's also using oxygen, and that's what we see in the middle of that formula there. And remember we talked about in these kind of reactions, you have reactants and then you have products of these reactions. And look at this product over here. What is that? Anyone know the name of that? H2SO4? Sulfuric acid. Okay. That sounds pretty bad. Well, I don't know if it's bad, but it's strong. It's a very strong acid. Okay, so on a pH scale, strong acid means what? Yeah, it's like very low number, right, on the pH scale, right? Okay, so we actually have some bacteria that actually likes that. So we have Acidithiobacillus. So you have Acidithiobacillus. And you may have seen this kind of orange coloration in water in our area because our area has a lot of iron. And if you go out west where you have the Great Rocky Mountains, there's a lot of minerals and ores that would normally be in the deep in the ground that are actually lifted up in the form of these mountain ranges and are therefore more accessible. So in that kind of environment, you have a lot of mining for minerals. And so, you know, one thing that is often done in these mining operations is that they use water to separate out the minerals from each other. So a lot of times there's these pools of, you know, leftover water with you know, basically minerals in them. So if those minerals contain sulfur, then we see the acetylbacillus growing in it, and it can actually release, you know, this sulfuric acid into these pools of water, causing them to be very acidic. Yeah. So don't drink the water. All right, so that was a chemolithotroph. Now we're going to looking at some examples of chemoorganotrophs. Chemoorganotrophs, those that use or can use organic compounds as their energy source. And we're going to be looking at two different kinds. One is an obligate aerobe. Okay, what do we mean by obligate aerobe?
Requires oxygen.
They absolutely require oxygen. That's like you, right? Okay. Now remember when we're doing our thioglycolate test for your bacteria. So in that test tube that you inoculated on Tuesday, you know, some of you looked at the results on Wednesday, but for an obligate aerobe, where would you expect to see your bacteria growing?
Near the top.
Near the top, excellent. So they like to grow at the top of that liquid where there's ample oxygen. Okay. And why? Why do they need like oxygen? Well, they need it as an electron acceptor. So that's what receives the electrons, which are found in these H's, the hydrogens, and then they, as a result, they produce water. Okay, so let's look at some examples. All right, 4 different examples listed on the slide here. There are a lot more than 4, you know, that are known to exist. I just pulled some out for you here that were in the textbook. So the first one is at the top. It's called Micrococcus, and there's a picture of Micrococcus on the upper right. So what shape do you think these bacteria are? They're circular, right? Coccus. Okay. And look at that. It says that that this is a common lab contaminant. So in other words, it's very abundant in the air and in our environment. It's not necessarily. They're not harmful. You know, they're just there. So this kind of picture here may, you may have seen this growing in your Petri dishes here. So some of you have these yellowish colored bacteria that may be Micrococcus or something similar to it. Okay, the second example on the slide is called Mycobacterium. Mycobacterium, and this is of interest. They are bacillus, so they're rod shaped. They do not make the endospores, but they do have walls that are very difficult to stain. So as a result, there's actually a special stain that we're actually going to be doing next week in the laboratory. So next week we're going to be staining using a little bit of acid and high heat in order to try and get stain into the cell walls of cells if Mycobacterium is present. So Mycobacterium includes a couple of interesting, there's a couple of interesting species there. One's called tuberculosis, which is like a lung disease. Another one is called another one causes leprosy. So leprosy is the one that's famous in the Bible because of the work that Jesus did with the lepers. But the bacterium is actually interesting because it prefers to grow in cooler parts of the body. So like parts of the appendages, like fingers, toes, ears, nose, that kind of thing. So that's kind of interesting if you think about it. All right, third one on our list is Pseudomonas, and this is the one that was kind of featured in your textbook when they were examining the spread of bacteria in an operating room. Remember that one picture of the room that had the red stains on the table, on the floor, and all over the place? That was supposed to represent the bacterium Pseudomonas aeruginosa, which often infects burn patients. And Pseudomonas aeruginosa is famous because it makes kind of like this greenish pigment. There's other Pseudomonas strains that make other colored pigments. So some of you may have Pseudomonas growing in your petri dish. And then the last one is called thermus. And there's one called Thermus aquaticus that actually likes living in really hot waters. It's called a thermophile. You can actually find it out west. I don't know, have any of you been to Yellowstone and visited those geysers out there? Anyone go to Yellowstone? Not Jellystone, but Yellowstone. Anyone? No? No one? Okay. All right, put that on your bucket list of places to visit. Well, it's in these boiling pools of water. These are naturally boiling hot springs where you can actually find bacteria growing in there. One's called thermus. And in order to survive in those super hot temperatures, they have heat-stable enzymes. And I know some of you are in genetics. You're gonna be performing this. I don't know. Maybe you've already done it. Have any of you done the polymerase chain reaction in genetics? Okay, good. So the enzyme that's used in that reaction actually originated from this bacterium called Thermus aquaticus. Okay, so those were the obligate aerobes. Now we're looking at the facultative anaerobes. So what do we mean by facultative anaerobe? How do they differ from the obligate aerobes?
They're not obligate.
They're not obligate, but how else do they differ?
They can grow in oxygen environments or anaerobic environments.
That's right. So they actually, so in your blacklight tubes, remember how they grow? Yeah, they're growing throughout the entire tube. So you know, if you stab the bacteria down the length of the tube, they can grow near the bottom, but they can also grow at the top. They actually prefer growing with oxygen, so you might actually see more growth at the top and a little bit less towards the bottom. But they still can grow in oxygen-deprived environments. And those environments include our intestines. So there are some what are referred to as normal microbiota. Okay. Anyone know what they mean by that? Normal microbiota. No?
No. Okay, present. What's typically present?
Yeah, what's typically found in your body, in or on your body. So in this case, these are microbes that are found in normal, healthy individuals. So these are not pathogenic. Okay. Yes, even E. coli is growing in your intestines right now. But those are not the harmful strains. You know, some of these microbes are actually really important for normal metabolism, for your normal metabolism. Okay, but then we have some bacteria that are harmful. So we have some of those, you know, we have E. coli on the list. You've probably heard of Salmonella. And we also have Shigella. Shigella is actually the source of the toxin that the harmful E. coli strains have and produce. The Shiga toxin, that's the one that's responsible for foodborne illnesses there. Okay, so they cause diarrhea, and I know a lot of times people don't really understand, you know, how bad that is. Well, I think it's pretty bad, but the idea is that you can actually die. You can actually die from that because you just become so dehydrated. Okay. So this is it's actually very serious. Other life-threatening diseases include, look at this one, Yersinia pestis. Yersinia pestis. Have you heard of the bubonic plague? Yes? Okay, good. So apparently back in the Middle Ages, approximately 1/4 of the European population is estimated to have died from bubonic plague. 1/4. Oh my gosh. Can you imagine that? 1 in every 4 people died? I mean, you thought COVID was bad, right? But 1 in 4 from the bubonic plague. And that's caused by a bacterium that was actually spread from fleas. From, anyone know from where? Yes, infected rats, that's right. So you have infected rats with Yersinia pestis. The fleas transfer the bacterium when they bite humans. So 1/4 of the population died. I mean, conditions must have been pretty tough back then, where there's all these rats and fleas. You know? That's the kind of world that they were living in. I don't know, just for fun, have any of you ever been bitten by fleas? Yes, I have too. It's like you just don't know where you might, sleep one night and then you end up being kind of bit by his fleas. So yeah, it can happen. All right. So the other items on this list here is that these kind of facultative anaerobes, because they can live in oxygen-deprived environments, they can ferment. They can undergo fermentation, and some of them prefer to ferment glucose. Others prefer to ferment another sugar called lactose, and you can actually use that as a basis to discriminate and identify one group from another. The coliforms, which includes E. coli, the ones that like to or can live in the intestines here, the colon, those prefer to ferment lactose. So we're actually going to do, next week, we're going to do fermentation tests with your bacteria to see, can they ferment? And if they can they ferment glucose or can they ferment lactose? Okay, so try and remember, we're going to be doing that one too next week. Okay, so what does that mean if your bacteria can ferment? For example, let's say it's a facultative anaerobe, because your bacteria grows throughout the thioglycolate tube. And let's just say that it does ferment lactose. So where does that bacterium come from? Someone's intestine. Okay. And we can talk about how it may landed from there to where it's in your petri dish, today. Like we can talk about that whole process, but we'll get there. All right, next we're going to look at more examples of diversity. This time we're looking at diversity in terms of where the bacteria prefer to grow. Where do they like to grow? So we're looking at, on this slide, examples of microbes that grow in terrestrial environments. What do we mean by terrestrial? Land, that's right, land. Soil, for example. You are a terrestrial organism. So we have some examples of bacteria like Bacillus and Clostridium. They are the ones that can make the endospores. So here's an example on the upper right hand corner. This isn't necessarily stained the same way that you did your stains, but you can kind of see there's a dark purple bluish outline of a cell. You see that? And then inside is that pink oval. So that pink oval inside of the darker purple, the pink oval is the endospore. So I think with the stain that you were doing with the malachite green, instead of pink, it would have been the green. So that's what we were looking for, like the green inside of the pink. So we didn't see that, so that's a good thing. we don't want Bacillus or Clostridium growing inside of our petri dishes. Well, why not? Well, that's because some of these are associated with some pretty nasty diseases. So last time I mentioned the Clostridium tetani, it produces that neurotoxin that causes the involuntary muscle contraction, where people were literally breaking their backs because of that. Like, we don't want that. But here's one I haven't talked about yet. This one's Bacillus anthracis. Bacillus anthracis. which causes a disease called anthrax. And that one's actually kind of famous, not because it's very common, but because around 2001, there was this bioterrorism event. About 7 or 10 people were actually mailed. They got a letter in the mail, an envelope in the mail, containing this white powder. And it turned out that white powder was the Bacillus endospores. It's like, what the? But they didn't know that. It was just this white powder. But they came down with anthrax, and some of them actually died. And it's not exactly clear who did it, but they did. One person was arrested and incarcerated. It was a scientist that was actually working for the U.S. government in their bioweapons department. So, like the U.S. government, as part of its military operations, does have individuals who have and are working with these what are referred to as bioweapons, so biological warfare weapons. Okay, so for some reason, if he actually did it, I don't know why, but he was actually using those as a weapon. Okay. Let's look at some other examples here of terrestrial microbes here. Again, these are the ones that prefer to live on land. And here's an interesting one called Streptomyces. So this one is a bacterium, but it looks like a fungus. So we've already looked at an example of a bacterium that looks like algae. This one looks like a fungus. So you can kind of see on the right that it forms these chains that look like hyphae, fungal hyphae, and mycelium. It kind of looks fuzzy when it grows. And this one I thought was interesting for you because it is the source of some very helpful, beneficial antibiotics. So a lot of times we learn about penicillium and how that's a source of penicillin. Penicillium is a fungus, but here we have a bacterium that makes antibiotics that kill other bacteria, like streptomycin, tetracycline, erythromycin. Do some of those sound familiar to you? Have you been treated with those? Yeah, those are very common antibiotics, and you have streptomyces to thank for that. All right, so let's think about that. Why? Why would streptomyces make these kind of chemicals? It's not making them for our benefit. How does streptomyces benefit by making these chemicals? What do you think?
Knocks out competing bacteria.
That's exactly right. It's a dog-eat-dog world out there, right? Very competitive. Streptomyces is killing out the neighboring competition, right? In order to acquire more food and resources. All right, let's look at another example here. Again, this is a terrestrial bacterium. This one's called agrobacterium, and some of you who are in genetics may have heard of this one already too, because this one, again, it's a bacterium but it can infect the stem of certain plants, and when it does so, it will transfer some of its own DNA into the plant cell. So this is a form of natural transformation. And then that plant will start producing hormones, growth hormones. So the plant cells start to divide, and when they do, they form a plant mass called a tumor. So that's what's pictured here on the right. Is this is very similar to cancer. The tumor is very similar to cancer there, and it's caused because of the plant was infected by this bacterium called agrobacterium. So have you learned about agrobacterium and genetics yet? Okay, I'm sure this one's coming up, so put this one in the back of your mind too here. So like in genetics today, people actually use agrobacterium to not only transfer its DNA into plants, but other kind of genes into plants to make them for biotechnology purposes. Okay, and then one more example here. You may have heard of this one too. Again, we're looking at bacteria that grow in the terrestrial environment. This one's called rhizobium, and then rhizobia is like plural. So these are bacteria that can infect the roots of legumes. Anyone know what kind of plant is a legume? Peas, yeah, beans, yeah, like soybean, for example. And then when they infect so this is like a root here, underneath the soil. And then when they infect the root, it'll cause the root to swell up, form this nodule, and inside of the nodule is where the rhizobia reside. And inside the nodule, they will fix nitrogen. So they take nitrogen gas that's in the air pockets in the soil, and then they'll convert those to nitrates and nitrites, which are actually usable by the plant as a source of nitrogen. So nitrogen is really important for plants because nitrogen is an important component of chlorophyll, that green pigment that plants need for photosynthesis. So when you see these plants with this symbiotic relationship where they're able to get more nitrogen, the plant leaves are more green than those that don't have the bacterium. All right, other than legumes, one other plant that rhizobia can form root nodules on is called clover. You've heard clover, right? I mean, March is coming up. Yeah, for these clovers, everyone. Okay. Well, clover is interestingly, if you look back in time, when people, had lawns in front of around their homes, they didn't always grow grass. What was more common is that they grew clover, and clover grows very low to the ground, so they didn't need to mow it like they have to mow grass today. And because clover has an association with rhizobia, it forms root nodules that can take atmospheric nitrogen and convert it into nitrates that can be used to make chlorophyll. Clover remains green throughout the year. You don't need to add fertilizer to it like you do with lawn grasses. Okay, so people used to have lawns with with clover that they did not need to mow, they did not need to fertilize. So, why are we growing grass now everywhere? Well, it's kind of sad, but the chemical fertilizer industry told people, hey, why don't you plant grass? And hey, while you're doing that, why don't you purchase our fertilizers? And everyone did. Can you believe that? Okay, so I guess it's a lesson where, just because you see the world the way that the world is today, it doesn't mean that the world has to be that way. All right, come on. I think we're actually going to wrap up here with that thought. And we'll pick up more with this one on Monday. One last thing I forgot to mention at the beginning. I got distracted by the lack of airflow here. But remember, we're getting ready for our next test on, which is scheduled on Friday of the coming week. I did post a practice test. I posted a practice test on our Blackboard site because it always, I think it's always helpful to plan ahead. Start reviewing for the test, even though we're still working on Chapter 11. So you can start working on that practice test, if you want to this weekend. And, give yourself time to prepare. Always prepare for success. Okay. Have a great weekend, everyone.